and when you treat it with, when you misexpress sonic, either by retinoic acid or on other means with beads and so forth, you get a mirror image duplication, just like a chicken. So the idea here is, and there's other evidence here showing linkages between the AER and the ZPA, which I'm not showing you, that really shows that this appendage patterning system with ZPA, AER, and all these other things really is very general to all vertebrate appendages, not just limbs. Which takes us to a much deeper question, which really is, how did vertebrate appendages arise? How did that whole signaling system come about? And what could that possibly tell us about the fossil record? The first question we really asked in this is, how do fins differ from other outgrowths in their development? And again, looking at whether it has ZPA and AER in different parts of the body. So we looked at sharks as an extinct one, but if you look at a shark or a skate, what you see is there are many areas that may be little outgrowths that could develop like a limb. Most notable of these is in the gill skeleton itself, and this is has some historical resonance. What you see here is a shark head in lateral view. This is the head, and this is the gill arch skeleton. If you look at this, it's cleared and stained. What you'll see is they're projecting off of each gill bar are these rays. And if you dissect them out, as Andrew Gillis, who was a graduate student in my lab, did, what you see is this is the gill bars here, these are here, and each ray sticks off the gill bar. The idea is that perhaps this little gill area here is like its own little fin. That is, these rays grow out in a fold of tissue, okay, extending from these bars. And this actually has some historical resonance in that a great anatomist, a comparative anatomist, Carl Gegenbauer, originally looked at these and compared them actually to fins and limbs. So the idea was to look for AER and ZPA in these things. Well, you remember that AER is this patch of tissue, it's a strip of tissue in the distal region of the limb that has FGF8 expression. We see it expressed in the skate fin, and surprisingly in the arch. These are very dark in this projecting dark. But what you have is a strip of FGF8 expression here, and when you do a section, it has a pseudostratified epithelium and behaves very much like an AER experimentally. If we look at these gill areas and we ask the question, does it have a ZPA-like area? Well, in the ZPA, you have a posterior zone of sonic hedgehog expression. When we look at the skate arches, what do we see? This is a ventral view of it. Here's the fin. There's the expression in the fins. But in each arch, in the posterior portion of the arch, you have a strip of sonic hedgehog expression and the receptor patched, which is there. Does it behave like a limb when we misexpress sonic and in a variety of ways? And the answer is yes. What we do is you could treat it with retinoic acid or insert beads or what have you. In a chicken limb, remember, when you treat them with retinoic acid, you get a misexpression of sonic on the opposite side, and it results in a duplication of the limb. Well, in these arches, you get normal expression, then you actually get you can see it here. There's a duplicated expression in that arch. You remember it produced a duplication in the appendage. What happens in the arch? You get a duplication. This is the wild type normal one. It's simply a bar that bends anteriorly. In the treated specimens, you get both the bar that bends anteriorly and a bar that bends posteriorly. So what's emerging here is that if you take a gill and its gill arch, a fin and a limb, there are profound similarities, and I've just covered a few of them, in the toolkit and signaling systems that pattern them. That when we look at a phylogenetically relevant set of samples, what we see is they're elements of the same genes, same signaling systems, and same functional effects in each of these appendages, from the AER to the ZPA. The question then becomes, what does this tell us about morphology? What does this suggest about expeditions targeted to the key, to key parts of the tree of life? Well, the obvious proposal would be that perhaps the developmental mechanisms that pattern appendages originally arose in another outgrowth. Perhaps that outgrowth was a gill arch. Perhaps that outgrowth was some other one we don't know about. The problem is when we look at the fossils we have and plot them on a stratigraphic record, we don't know a whole lot. There's a huge gap in our knowledge from basically the Wentlock of the Silurian all the way through the Middle Ordovician. We would predict, from a knowledge of development, that there should be non-limb outgrowths present in these jawless fish that are present from the Silurian to Ordovician. The challenge is we really don't know anything about them because no one has ever done targeted collecting of vertebrates to these sites. And in fact, I like to think that if in 40 years there will be people, the graduate students who are in the back, as Eric Lander was saying, who are doing genomic research, I'd like to think that there are a number of graduate students who will have found Ordovician vertebrates that tell us something about the outgrowths that are likely primitive to fins. So really that's what we're doing at our next set of expeditions, are actually to move much deeper in time to begin to look at these nodes here, which are very poorly understood in the fossil record. And armed with our knowledge of development, we are expecting an enormous number of non-limb outgrowths to be established. There is some comfort to be had here in a work by Philippe Janvier in 2008, where he described a little tiny jawless fish, Euphanerops, which is this shmoo up here. But when he showed it in detail in his paper, it is very clear that this thing has little gill rays. A jawless fish with gill rays. We've only known them in sharks before. So this is suggestive evidence that perhaps early jawless fish did have this. But our goal in terms of the expeditions we're going to lead is to see whether this kind of pattern is actually present all the way through the basal portions of the tree of life, in the common ancestor of all jawed vertebrates. All vertebrates. So basically what I want to say is that we can use the fossil record to make targeted expeditions and to make targeted discoveries in key nodes in the history of life. Those fossil discoveries give us new ways to think about the anatomy and the development of extant creatures. And the more we study extant creatures, the more we have ways of reinterpreting the kinds of morphologies that may have existed in the distant past. This is a wonderful reciprocal illumination between different areas, different approaches to understanding the history of life. And the only way that they are united is through the profound power of descent with modification and the tree of life that's resulted from it. Thank you very much. Yeah, they don't look like paired ventrolateral fins to me. Yeah. But what we do know, though, is that so when you look at what we can infer is that paired appendages are relatively recent. Unpaired appendages, particularly a median fin, is present first. And then what we're proposing here is likely here, is that the gills and gill rays are actually the original system that established those. Yes.
Where is the expedition going to be?
Yeah, so, sorry about that. Much to my family's dismay, it's up in the Arctic. There is an enormous amount of Silurian and Ordovician in the Canadian Arctic, which I've flown over for years, actually flying over it, looking northern Baffin Island, Somerset Island, Cornwallis Island. So I've looked at it. I never realized I'd be actually proposing to work there. It's very well exposed. It's nearshore marine, and it's never been worked by anybody.
What's known as developmental mechanisms of arthropod?
Quite a bit, yeah.
All related to this?
Well, they have scary similarities to some of this. So versions of the same genes are doing sort of analogous things. And so if you look at hedgehogs and what they're doing in there, if you compare some of the genes, one gene called DPP compared to BMP4 in vertebrates, there are stunning parallels that are analogous in many ways. The real question is, does this extend deep down to arthropods? And it may well be that what's happening is here is the information is being carried along in either outgrowths or some other organs that we need that use these signaling systems. The vertebrate pattern seen both in the gill arches and in the limb is different in a lot of ways. I mean, it has some uniqueness to it. What we're actually taking is another approach, which I didn't talk about, is there should be a regulatory signal to this as well in terms of the tissue specificity of the cis-regulatory elements that are, some of them that are behind this as well. So that's another way of approaching it. Okay, well, thank you very much. I will introduce